Hi, welcome to Fitness Facts. In this video, I'm going to cover how a muscle contracts, so you can better understand things like fatigue, muscle soreness, how to train for strength versus size, just to mention a few. My goal is to provide enough easy to follow information so you can understand the process well enough to apply it to your own exercise routine or training program. I won't be able to get to everything, so leave your questions and comments in the comment section below, and I'll get to them as soon as I can. Muscles are designed to do one thing, and that is to shorten. In other words, to pull one end of the muscle towards the other, in the process causing movement at the skeletal joint that they cross over. This shortening occurs when thin and thick protein filaments inside the muscle fiber or cell interact in a sequence of events called the sliding filament theory. This happens somewhat separately in the thousands of muscle fibers that make up a muscle, which I'll explain as it happens in a single muscle fiber. It all starts at the end of a motor neuron when it releases the neurotransmitter acetylcholine, which moves across the cleft or gap between the neuron and muscle fiber. Here, acetylcholine binds to receptors on the sarcolemma, the membrane of the muscle fiber. When enough acetylcholine binds to the sarcolemma, the membrane of the muscle fiber, it opens up channels that allow sodium to move inward and potassium to move outward, producing a small electrical current known as an action potential. The acetylcholine is broken down and removed, but the action potential continues to spread like ripples in a pond of water. As one sodium moves inward, it causes neighboring sodiums to do the same, over and over until the action potential spreads over the entire sarcolemma. Throughout the sarcolemma are small holes or openings called transverse or t-tubules that lead inside the muscle fiber. As the action potential spreads, it also goes down the t-tubules, which is surrounded by calcium storage compartments called the sarcoplasmic reticulum. Inside the t-tubule, the action potential activates receptors that are connected to channels or gates on the sarcoplasmic reticulum. These channels open up, allowing the calcium to escape and spread throughout the sarcoplasm, the fluid that fills the inside of the muscle fiber. This ends what is referred to as the excitation phase and leads us into the contraction phase. The calcium ions bind a part of the thin protein filament called troponin, one of three protein molecules that make up the thin filament. This causes a second protein called tropomyosin to shift or move, which exposes the third protein called actin. The actin protein has binding sites on it, where the heads or extensions of a separate thick filament called myosin can now strongly attach. Now up to this point, the muscle hasn't even contracted, but once that connection between the myosin head and the actin is established, the myosin head swivels, pulling the entire thin filament inward, and then it releases from the actin. As long as acetylcholine continues to be released, and eventually that calcium gets released, the myosin head will reattach, swivel, and then unattach over and over again. And that ends the contraction phase. The muscle fiber is organized in small sections called sarcomeres, where the thick and thin filaments reside. During contraction, the thin filaments on the left and the right slide inward towards each other, over the thick filaments which stay put. The collective shortening of millions of sarcomeres and thousands of fibers cause the entire muscle to shorten. So when enough fibers in a given muscle shorten or produce enough force, the entire muscle contracts. Let's look next at how the muscle relaxes. The first step in muscle relaxation is for acetylcholine to stop being released. Once that happens, the sodium and potassium on the sarcolemma are pumped back inside and outside respectively, and calcium gets pumped back into the sarcoplasmic reticulum. Tropomyosin shifts back, covering the binding sites on the actin, preventing further myosin head attachments. This allows the muscle to relax. Now lastly, let's look at the role ATP plays in muscle contraction and relaxation. Energy in the form of ATP is needed for those myosin heads to swivel. It starts when ATP binds to a myosin head that is just swiveled but remains attached to the actin. The ATP causes the myosin head to break away or separate from the actin. Then the ATP remains on the myosin head, where it's broken down by an enzyme called ATPase. This releases ATP's energy where it's stored in the myosin head, leaving it charged and ready to swivel the next time it binds to actin. ATP also fuels the sodium and potassium pump and the calcium pump as part of the relaxation process. And there you have it, the excitation, contraction, and relaxation of a muscle. Now here are some application points. Understanding this whole process will help you appreciate the difference between fast and slow twitch muscle fibers, which I cover in another video. It will also help you see the difference between sarcoplasmic and myofibrillar hypertrophy, or training for muscle size versus strength. And you'll have a better feel for what actually happens during muscle fatigue and soreness, and what's needed for muscle recovery, including the role of satellite cells. I hope this helped, and let me know if you have any questions, comments, or suggestions on the structure, function, and result of exercising muscles. As always, my answers come from peer-reviewed research linked in the comments section.